Hello, everyone, and welcome to Pocket Size Bio with Dr. Casco. In this video, we're going to take a look at Chapter 8 of the Nelson WACE biology textbook, which is Evidence for Theory of Evolution. If you follow the videos, we've gone from DNA and genetics and inheritance patterns, and now we're going to jump into evolutionary theory and some lines of evidence to support evolutionary theory, right? So let's get to it. So Chapter 8 essentially starts with Section 8.1, of course, living things change and diversify. So I've mentioned this to you guys in the classroom already, but I'm just going to go along and cover everything from the beginning. Right, there are some things that I've will omit that I've seen that they don't have really. I've noticed that they aren't present in any kind of assessment there. All right. So, when you talk about, living things and they change and diversify, there's a couple of concepts that you need to kind of familiarize yourself with. One, of course, being time. It takes a lot of time for things to change, right? In the whole scope of biology. So, when you think about how long life has come about, right? The amount of time it took life to even start and diversify, it has to be quite long, right? So scientists have kind of estimated that the life has existed on Earth for approximately 3.5 billion years, right? And ever since that inception of a fully single-celled organism, it's been changing over time, right? And all the way to this present moment, right? Living organisms are still changing, right? They're still coping with changes in the environment and adapting and things like that, right? And ultimately, that's the whole idea of evolution, right? As an organism changes over time, it develops characteristics that enable it to survive in a particular environment, right? And it does take a while, right? We're going to take a look at that. And yeah, it does take a while to get to the point where the organism has the features suitable to thrive in its environment, right? As much as it can, as efficiently as it can, right? So when you think about diversity, it's because those organisms have gone through a huge amount of change, right, through time. Okay. And one thing that we need to emphasize on is that these changes occur at a population level, right? Over long periods of times. And if you were to get more specifically, get into this notion, we are referring to genes, right? Genes are what create that change in phenotype, because ultimately what gives us our trait are genes, right? So all these changes that are occurring through a population over a long period of times are changes in genes, right? And it's those genes that, of course, are inheritable, right? They get inherited, right? They're what ultimately changes the population, which changes the gene pool, right? That, in a nutshell, is evolution, right? So evolution is a process of cumulative inheritable changes, right? In a population over many, many generations, right? Over many generations. Sorry. And ultimately scientists and biologists, they've kind of come to this task of trying to make a whole collection, and catalog of all the diversity of life and see how you can compare all these living organisms, right? So there's a collaboration between Australian museums, herbariums, universities, governmental agencies, CERO, um, and so forth, that started in 2006 as a project to kind of assess that biodiversity information and get a sense of some form of classification and evolutionary relationships, right? So definitely it's critical for Australia to be part of it because of Australia's unique diversity that we've seen in Unit 1, right? So definitely they have some unique, native organisms, right? And by cataloging all these organisms, you can get some better evidence of evolution, right? So, um, getting into that, the actual term of evolution, right? Evolution means to develop gradually, right? To change gradually. And again, one thing that I've need to note is that organisms themselves don't evolve. They moreover they acclimate, right? They can get some change in their physiology, but it's temporary. It isn't something permanent, right? Um, organisms themselves cannot evolve, right? Because um, you need to pass down heritable information from parent to offspring, not the individual itself, right? And so forth and so forth. So when you pass enough genes and enough variations of these genes in a pool, and those genes are enabling the species to succeed better, this is what would occur is that you'll get a new species, right? And so forth. Now, the concept of evolution has um, changed throughout history, right? I'm just gonna briefly go over this, right? Um, uh, for you guys to kind of know, right? But before I get into a little bit of history of evolutionary thought, you know, evolution can be seen at a macro scale or a micro scale. And we're going to see this in chapter 9 more in detail. But, it could be as subtle as having a little protein variation, creating different blood types, or it could be a huge change in even organism groups, right? Classification groups, right? Um, macro evolution. Okay. And we'll see this more in detail in the next section, in the next chapter. So just a little bit about a history of on evolutionary thought, right? So as you guys know, or probably have heard of, you have Darwin and Wallace being the key players on the idea of evolution, but there were some notions of change before, right? In the 1600s, you had this idea of natural theology, right? But it was more a philosophy rather than a biology there, where um, you have that idea that every kind of organism has essential, unalterable characteristics, right? So that's already one key thing about evolution: features, right? Characteristics, uh, adaptations, right? But as biology changed, as you're getting scientists, right? Observations, more notations, right? Um, Scientists started observing, right, more variation between different species, right? And that gave the idea of change in form, right? And then with the discovery of fossils, right, um, you have something to compare to in the past. So fossils, you know, um, generally are very different from present organisms, right? So you have the idea that you have this prehistoric form of a particular species and its current species, right? An ancestor in a current species, right? So you get this idea that there must have been something different, right, between the two. So that also gives the idea of extinction, right? Where you had a previous group of organisms. They're not present anymore, but you do have this newer form of an organism. So we'll get back into this in a bit, but you do need a form of change where some organisms die and some of them survive, right? Um, there's a famous um, a scientist, John Baptist Lamarck, right? And he had this idea, this theory of transmutation of species, that organisms pass on characteristics that they acquire during their life, right? So, and this concept drives adaptation, which ultimately drives change, right? So, you have this little Lamarck's giraffe, that you have this ancestral short neck giraffe, and that little giraffe. would stretch its neck as much as it could, and then that constant stretching will lead to an offspring that would have much longer um, necks, and so forth and so forth and so forth, that um, it will develop this um, um, you know descendant that would ultimately get um, a longer neck, right? Um, theory of transmutation, right? But Lamarck. Another example would be um, let's say you're working out, right? You're building your muscle, you're at peak, you have all these, you have a muscular body, all these gains here, and then you have a kid. He instantaneously, naturally would inherit this muscular body, right? Just because you worked out a lot. So that would be another example of theory of transmutation, right? Of course, that doesn't happen. Um, but I don't want to say he's completely inaccurate, because now things like epigenetics can give some form of support for Lamarck, right? But then you get these guys, theory of evolution by natural selection, 1800s, right? Mid 1800s, you get Charles Darwin and Alfred Wallace. Um, yeah, these two naturalists, and they would collect, species in their travels, right? And uh, moreover, Darwin had the better means to be moving around, and he essentially collected thousands of samples of organisms, particularly in the Galapagos Islands, and he he noticed many organisms like the finches, like turtles, the Galapagos turtles, tortoises, right? Um, and so forth, that ultimately led to this writing of a theory, right, which he basically wrote down in a book called on the Origin of Species, right, by means of natural selection. So he kind of stapled this process called natural selection. And natural selection is basically the process in which evolution works, right? And we're going to get into that in a little bit, where it's basically that the mechanism, right, or the process, like I mentioned before, in which evolution is driven, right? And overall, this theory that he's postulating, that organisms change over time, um, also consists of having a common ancestor, right? So this idea of change basically links all organisms to a common ancestor, right? And that we kind of know now by evidence of DNA and RNA and protein, you know, um, omics, right? So there's a lot of substantial evidence that can support that, right? So, what are some of the bases of Darwin's theory of evolution? Is basically, within a population, you have variations within a particular population, right? And those variations that are preferable would most likely survive, and then those organisms will pass those genes on to the next generation. Those that don't have preferable variation would basically die, right? Die out from whatever X pressure that's in the environment there, right? And we're going to take a look at some examples. Um, I mentioned the snails before already, right? And then those variations that survive pass down, and then the trait becomes more common. Overall, many generations pass down, that becomes the staple trait, right? So this is, in a nutshell, what evolution is by natural selection, right? It's also called descent with modification. And um, Darwin also proposed this concept, right, of a tree-like scenario where species would ramify from a common ancestral species, right? So he had some idea of phylogeny, right? If you guys remember, phylogeny is that the concept in which you have um, uh, different organisms ramifying from a common ancestor over time, where you can see similarities and differences, right? We're going to come back to phylogeny in this chapter, right? So this is just a general intro about evolution. We're going to talk in more detail about the lines of evidence in which evolution is supported by, right? That you can see here. Biogeography, comparative genomics, the fossil record, comparative embryology and anatomy, and so forth, right? So section 8.2 is again that same concept of life has changed and diversified over time. So I didn't mention this in the class, but uh, because of fossils, well, we're all familiar with what fossils are, right? The remains of previous organisms or ancient organisms. You have a collection, right? Once you are able to collect so many different fossils all over the world, right? You can create a geologic time scale or like a ruler, right? Um, or a fossil record of um of how life has changed over time, right? It's called the geologic timeline that essentially depicts, major changes that have occurred through the history of life, right? And scientists break this kind of timeline into several sections, right? Um, and it spans from the the beginnings of the earth, right? The birth of the earth and until now, right? And just as we kind of break down a year into months, into weeks, into days, and to so forth and so forth, you have this timeline that we break down into the broadest division, which is eons. From eons, you have several eras, and then within each era, you can have several periods, and then within each period, you can have several epochs, right? So the broadest division is an aeon. The most specific division is a epoch, right? Among these, you probably should know the aeons and the eras. Okay. Periods might be a little bit too specific for you guys. All right. So, uh, so what has happened in a nutshell, right? You have um, scientists collecting, paleontologists, archaeologists, they're collecting fossil samples and from everywhere around the world, and they're collecting different samples and different layers of the earth, right? And you start to get a sense of how, depending how old the fossil is, you get an idea of what type of organism existed at that particular time, right? Because of that, you can make this timeline, right? And you have these aeons, right? So the most prehistoric aeon is that Hadean era. Sorry, aeon. Then Archaea. There's Archaean era, aeon. Sorry, and then a Proterozoic, and then you have this Phanerozoic, which we're currently in, right? Eon. And then within this Phanerozoic aeon, right? You get into the eras that you've probably are familiar with from elementary school. Early Earth, Hadean, right? Basically, formation of the Earth. Nothing is forming here. As time is passing, remember 3.5 billion years ago, you're starting to get the Earth cooling enough where you can get some little microscopic, even molecules, organic molecules forming. Um, Proterozoic era, eon, right? Um, you're getting very simple, very, very simple organisms, right? Bacteria, photosynthetic organisms like algae, some jellyfish, right? And then you're getting the Paleozoic era, which means paleo, meaning prehistoric life, where you're getting pretty much amphibians, fish, a lot of aquatic organisms, um, some simple types of plants, right? Ferns, and so forth, ultimately transitioning into that Mesozoic. Meso meaning middle, right? It's also the age of reptiles. Basically, this is the age of the dinosaurs, where you're getting the Triassic, Jurassic, and Cretaceous periods, right? And then the Cenozoic. Cenozoic meaning the modern, right? Or more recent life. Okay, here's where you get the age of mammals, basically, right? Birds and mammals and so forth, right? So in a nutshell, this is a what the geologic timeline is about, based majorly on fossils, right? So related to this is the concept of continental drift, if you remember from Earth science, right? And in order to get a sense of this whole idea of fossil record and where the fossils are um throughout the layers of the earth, you have to understand a concept of continental drift, right? Where you have earth that has been the surface of the earth is always changing. It's changing now, but it's such a slow thing that you really don't notice it on a day-to-day basis. But when you have a span of millions of years um in terms of time, definitely there's some massive changes happening to the Earth's surface, right? So the continents that we think about today weren't the same as how they are now to how they were in the past, right? Before you had this supercontinent called Pangaea, split in half. You have two continents, Laurasia and Gondwana, right? As time is passing, you have this Triassic era, period 200 million years ago, 135 million years ago, even more, subdivisions in the continents, as plates are moving throughout time, until we get where we are, right? So, um, understanding this idea, we're going to get into in a little bit with biogeography, right? But definitely there's a correlation between, geology and evolution in biology, right? Continental drift. One thing related to this is so I kind of mentioned this already. So definitely with the tables, I'll tell you guys to take a look, right? At these tables here. But let me go up a little bit. But within this geologic timeline, scientists were able to infer that some events happen, throughout time, particularly extinctions, right? Scientists, if you remember from Unit 1, I kind of showed you this ice core um, a sample that scientists can deduce levels, how the environment was depending on the bubbles and the chemicals that were stored in these frozen ice cores, right? So um, you can get a sense of how the atmosphere was based on, these samples, right? So, climate, sea level, atmospheric oxygen levels could be analyzed. So scientists were able to deduce that Earth's climate fluctuated, right, between very hot and humid periods to very cold and dry periods, right? And by analyzing these ice cores. So, um, yeah, very interestingly, throughout Earth's history, it was, much, much warmer than what we think about how it is now, right? So, and in the past, the temperature difference between the equator and to the poles, latitude wise, wasn't so different, right? So it's quite interesting how warm, how uniform it could be, right? In terms of the Precambrian era, right? Around 12,000 years ago, you have these two periods, right? The Carboniferous and the Permian periods, where you would have a lot of ice, right? It would be very cold. Yet snow glacier sheets covered much of the earth, right? Not just the poles, but around everywhere, right? And then in between these periods, you will have more tropical climate, right? Higher temperatures and so forth. So you have a fluctuation of climate over a range of years, right? So what fluctuated? Basically the atmosphere. Atmospheric conditions fluctuated, and there were critical factors to sustain life. Of course, you need to have a very stable environment to be able to kind of nurture life in general, right? You have plants and you have the herbivores that are able to sustain themselves by eating plants and things like that. So, um, conditions needed to be just right. Okay. If we go back to the beginning, early Earth almost had no oxygen, right? No atmospheric oxygen. Okay. But these levels began to increase due around 2.5 billion years ago due to photosynthetic bacteria, right? That were able to capture carbon dioxide, which was present more, right? Where you can get photosynthesis occurring, you can jumpstart, organic compound synthesis. So that enabled a whole metabolic movement, right? Where it would lead to the growth and diversification of eukaryotes, right? So you had this idea of, atmospheric oxygen formation leading to a kind of boom in diversity, right? Even if it's single celled. Okay. So understanding that these concepts, we can get an idea or a sense of these different lines of evidence, right? Um, you have biogeography, where it basically this concept talks about distribution of organisms, right, and ecosystems across the world, right, through geologic time. Okay, through geologic time. So I'm just gonna move a little bit higher here. And what's interesting about Australia is that, you know, because they're an isolated landmass, they do have a uniqueness um, uh, in diversity, right? They kind of separated early on, so they were able to diversify themselves in a particular way, right? But nevertheless, when all the land masses were together, they did share some similar plant and animal groups, right? Um, and different continents would have species that spanned across all the continents when it was Pangea, right? And one of the lines of evidence, or the reason for biogeography being a line of evidence, is that you can see, um, you know, whenever you had Pangea, you had these early organisms that would thrive throughout these different continents, right? And this is based on fossil evidence that scientists have accumulated, you know, over time. For example, this particular species here, and I'm just gonna zoom into that. I don't know why I left it so small for so long. Um, um, Cynognathus, right? Triassic land reptile. Scientists collected samples in Africa and in South America of this particular species. You had this particular fern here, Glossopteris, right? That would span all continents, right? Which is quite interesting. And the same thing for all these little other animals here. So that gave you a sense that evolutionary theory had some consistency with time because it took a long time for it to happen, and that the plates moved, right? So you had some strong evidence there that actually the Earth surface was actually circulating and moving around, right? Because or else how else could the um, organisms be in all these places that are so vast, right, and far apart, especially um, or animals if they're not connected, right? Okay. So fossil observations and current species distribution patterns, right, were made by biogeography. Okay, so that's quite interesting there. Okay. So getting into evidence of for the theory of evolution. So this is the first one in terms of the book order, right? Comparative genomics. We just finished this concept, so I won't go into so much detail with this, but uh, yeah. So this would be your kind of first major or your second major line of evidence, right? Comparative genomics. So, um, genomics, right? Omics, genome, right? It's basically the study of a whole set of genes of a particular species. And um, when you talk about comparative genomics, right, is basically you're comparing, you're analyzing genes from two different species, right, and see how they are similar or different, right? So genomics in itself is basically those whole set of genes within a species and how these genes work within a genome for the organism, right? And um uh from biotechnology lessons, we know already like sequencing and many organisms have been sequenced nowadays, humans, chimpanzees, koala, many tremendous amount of bacteria, the fruit fly, worms, frogs, fish, and mice, right? So as you can understand more and more, the molecular makeup of an organism, you can see how similar they are and how homologous they could be, right? Um, from one another, right? And ultimately, what are you comparing? Basically, you're comparing DNA, but you can also compare RNA and you compare protein structures and see to a very fine degree how related they are, right? So this is very powerful. It's a very powerful tool to see the degree of relatedness, right? So um uh by comparing genomics of different species, you can also talk about conserved genes, um uh, genes that are essential for DNA replication, they might be conserved well between many, many different forms of life because it's such an essential process to thrive as a living organism, right? That you need. So it's quite interesting, right? Um, how much is conserved, how much is similar, you can start making groups called taxa, right? Um, Basically, if you remember from Unit 1, the different taxa are the different levels of organization. You have these kingdoms and orders and things like that, right? Yeah, King Philip, if you remember. Okay, so within these taxa groups, you have these clades where we're going to do some practice problems here. So I'm going to get back. We're going to when we come back from the break, and I'll actually go over this in class. But just to give you the kind of notes here, um, within taxa, you talk about clades, and clades are groups of organisms, right? You refer to a group of organisms or a current species and the descendant, right? Sorry, an ancestor and all the descendants of that ancestor, right? And you use these clades kind of to group, uh, different organisms that you're referring to, right? And it helps to measure relatedness, right? Um, and there's different ways that you can make up these clades, and generally it's by DNA, right? So you have this idea of genomics, and then comparative genomics, like I mentioned before, you essentially have this idea of, comparing different species by sequence, right? So this is an area of a big area of trend right now of a lot of investigation, a lot of um, it's a hotspot in biology, right? So field of bio of comparative genomics is a field of biological research, right? Where researchers use a variety of tools, bioinformatical tools, moreover, to compare genome sequences of different species, right? So in a nutshell, essentially, the more similar the sequences are with each other, the more related they are in their evolutionary history, right? And the reason because of that is because less time has passed between each uh, species A or B that you're comparing to accumulate mutations. So um, the less time that has passed and they're very similar means that they must be closely related, right? So it does help, determine relationships, right? So by having these relationships, not just of gene sequences, but um, appearance, behavior, and biology, you can understand to what degree, they're related and how much each individual species has changed over time, right? So there's no staple measurement within sequencing. Um, you can use specific genes, exact sequence of a particular gene. You can have number of introns that repeat is another way to measure sequence similarity between species. You can compare an entire genome, but there's no real staple ruler, right, to on how to measure, right? So let's take it for example, the human and the chimp has 4% difference in terms of genomics, right? So one interesting thing related to that is that scientists can compare the rate of change between certain genes, right, among humans and chimps, right? So you can get a sense of um, how closely related we are. Again, like I was mentioning before, depending on how fast the mutations occur and how they accumulate on the genome, right? So it's referred to as rate of change, right? In uh mutation rates, if I can say. Okay. There's a little diagram that can depict, in a sense, how sequence similarity is, right? So you have certain common features. You have this particular gene that must be vital for all living organisms. So you see it that it's here, right? Depicted in yellow. Uh, yeah, you know, um, as you go along, you have these different variants of particular species, right? And how different they are. Um, as you're talking about different genes there, right? Um, yeah. So not so essential, this picture here. All right. DNA hybridization is a method to analyze relatedness, right? And very similar, to no, I don't want to say to the genomic methods we've seen so far. Um, one particular method is called DNA hybridization, right? Um, have been used to measure relatedness. So, what happens here is that you get DNA that is being extracted from two organisms. You purify it, right? You cut it into fragments. The DNA gets unwounded, so you basically break the hydrogen bonds there, and depending on how similar they are or how identical they are, the different species, their DNA could hybridize. Hybridize meaning that the species of the DNA of one species, let's say, for example, the flamingo, can link to the other half of the DNA with this particular bird, the grebe, right? Horned grebe. This can tell you a degree of relatedness, right? Indirectly, right? So essentially, this is what DNA hybridization is. Okay. And the same thing could occur with RNA, right? But it's the same premise, right? Resistance to separation can be measured to work out evolutionary relatedness. How stringent is it to how difficult is it to bond, and hybridize? Hybridize means basically to bond between different DNA templates. So by analyzing the variation of DNA or RNA sequences, scientists could obtain a measure, right, of the difference between organisms and trace evolutionary relatedness, right? Ribosomal RNA has been used extensively to measure evolutionary relatedness, right? Because it's a highly conserved, right? Every organism needs ribosome. How different is the ribosome is a different story, but every organism uses ribosomal RNA, right? So it's a good indicator to check relatedness, right? It's a highly conserved DNA and RNA sequence, right? Okay, so a method, right, to learn relatedness. Okay. Comparative genomics and several other omics studies also are based on what is called bioinformatics. This is a big, big trend, very important tool in biology in general, and you can get a whole career in bioinformatics, right? So basically, bioinformatics is the application of analyzing data, right? So all this DNA sequencing that I've been talking about is actually stored digitally, right? In databases you can find online, all these sequences you can find online. But that's just the tip of the iceberg. How you manipulate that data is is a whole story, right? So um definitely genomics produces large amounts of data. So petabytes, incredibly big, large amounts of data, right? You probably even have never heard of petabyte. It exceeds terabytes, right? So data can be stored and analyzed, right, in a logical way due to what is called bioinformatics, right? bioinformatics is basically it's become a discipline of biology where you combine, engineering techniques, computer science, mathematics, mathematical models, principles of biology all into one to essentially analyze these particular samples, right? These sequences. So in itself, bioinformatics is the digital storage, retrieval, organization, and analysis of large amounts of biological data, right? Primarily in the form of nucleotide sequences, DNA sequences, amino acid sequences, RNA sequences, right? Um, um, Structural biology, uh, modeling, right? So all of these together can give you a sense of a relatedness or output that you see here, for example, right? So, um, with the birth of bioinformatics, there's been a lot of progress in how much knowledge we know, how much understanding we know about relationships, right? About genes, right? About phenotypes between patients and normal wild type normal individuals. An example that you can see here is the PAX6 gene, right? PAX6 is a gene that that creates a protein that controls how the eye is formed in vertebrates. Okay. And it came about from an ancestral PAX like gene, right? Evolved somewhere here. And several variants of this gene has changed over time to different groups of life, right? Um, and Nevertheless, we have some similarity with different organisms. For example, 69% of this gene is similar to a protein that the octopus has in his eyes, right? So you can get a sense that um, this gene has changed over time through different groups, right, of life. So you get a sense of homology, right? Homology meaning similarity, right? So let's say here's the mollusca, here's where the octopus would be in their group, and we have the vertebrates here. Yeah, definitely a big extension there, but nevertheless, it's there, right? So whenever you analyze this type of information, you're doing basically molecular phylogeny, right? Molecular homologies can provide information to build phylogenetic trees, right? Just by simple sequence similarity, alignment, right? We're going to take a look at that also as a practical. Okay. In terms of phylogenetic trees, I'm going to teach this for you guys in the class. Okay, we're going to do some examples. We saw this in unit 1, but I'll get back to it in this unit. Right. So for now, I'm just going to jump a little bit on that. All right. Again, what I mentioned before about mutation rates, guys, nothing too detailed here, right? But it's essentially a way that you can measure a mutation rate and you can get some sense of similarity based on the rate of mutation, right? So the frequency of new mutations in a single gene or organism over time is generally pretty constant. That's something quite mind-blowing, right? The rate of which you get a mutation is quite constant, right? It's called the mutation rate. Mutation rates are actually they can be used as clockwork, right? To estimate when a certain species diverge from another species or ancestral species, right? Um, because of the uniformity in change, right? So that's quite mind-blowing there, right? Humans' mutation rate is estimated to be approximately 10 to 8 um, uh, changed nucleotides, right? Per base pair per generation, right? So not too much, if I can say. Not too much. All right. Similarly to what I was mentioning about the DNA sequences, you also have comparative biochemistry, right? Proteomics, comparative protein conservation, right? So again, very similar to DNA. You have this idea of organic compounds and amino acids, protein sequences. Remember, the sequence of DNA produces the sequence of amino acid, right? So um, by having this sequence similarity, right, you're able to get a sense of um, how different they are, how similar they are, right? So comparative biochemistry is the study of different kinds of proteins, amino acids, including enzymes, all any type of protein, right? And cell machinery. All right. So again, you could have it with DNA, where you have certain sequences of DNA, between different species. And here you see an example of the sequence of many, many different species where this particular area of the DNA is all conserved. Everything is identical. And then here on this side, almost identical except for this G here. But something must have happened evolutionary wise that some of these species added several sequences here, right? Could have been a little chromosomal flip there, a little section, a little segment, a little break and fusion of DNA sections, right? Little insertion there for these two guys, substitution for this guy, right? So um something must have happened that enabled them to have more sequence in this particular area. Nevertheless, they can conserve this particular areas here that are colored, right? So in itself, that can give you a sense of similarity, right? Uh, between different species. Okay. It doesn't mean that the whole protein change. Maybe it can still do the same function, right? But it just got some more DNA embedded on it, right? Again, similar with proteins, small number of differences indicate a recent divergence, right? Because you didn't have enough time for changes to occur. Large number of differences indicates a more distant evolutionary relationship. Okay. So again, we're going to come back to this when we go into phylogeny. So, for example, the chimpanzee, when compared to humans, right? You have the chimpanzee 0 of the amino acid sequence. Gorillas 1, gibbon 3, orangutan 4, macaque the 8, the lemur 5, right? So, um, subtle differences between different groups of primates compared to humans, right? Chimpanzee being the closest to us, and vice versa. All right. Good, 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 good. So I mentioned about the fossil record for you guys. This is definitely part of the task that I want you to study. Study this well, fossilization, um, how to make healthy fossils. There's different ways, okay, uh, and so forth. And I'll probably go through this. Okay. All right. I want to go over evidence for evolution, comparative embryology, right, and anatomy. And let's take a look here also. Types of evolution. Okay. Um, Evidence of evolution, comparative embryology, and anatomy. So I'll just talk about this very quickly and we'll be finished with this little session here. All right. So um, let me just kind of move this a little bit closer. All right. So in terms of embryology and anatomy, right? If you take a look at these pictures here, right? Casco quality. Yeah. Casco standard. How nice these pictures are. Which one, which one of these organisms do you think is closest related to humans, to us? Beep, 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 beep. All right. You have a jellyfish, you have a sponge, You have a beetle. I believe there's a Goliath beetle, or the Hercules beetle. Can't remember exactly which one. You have an octopus. You have a sea squirt. Which one do you think is closest to humans? All right. Is the answer here? No, it's not. The answer actually is the sea squirt. Look, it's transparent. How's that possible? How is that possible? The sea squirt is more related to us than an octopus, than a beetle, and so forth, because of how it develops, right? How it develops early on. So, comparative anatomy and embryology is a way that you can tell or talk about degrees of relatedness based on physical features of a particular species at the embryonic level or at the adult level, and kind of get a sense of how similar we are, right? Comparative anatomy, we're going to talk about in a little bit. Um, and basically, it studies similarities and differences in structure between organisms, right? Like, let's say, like bone structure, bone morphology, right? Bone organization. Structural features are also called morphological features. Okay. We're going to get into that in a little bit. Comparative embryology studies embryos, right? And how they develop and form, right? So I'm going to zoom in a little bit here, zoom out a little bit here. All right. Comparative embryology is pretty much used to describe evolutionary relationships, right? And common ancestry based on how the embryos develop, how they're formed, their structure, and how they're developing, right? Between different species, right? The longer embryos of different species are similar during development, right? The longer the embryos, so if you make sure you get that down, the longer the embryos of different species are similar. So as they're developing, okay, over time, let's say like 1 month, 2 months, 3 months, and they look the same still, it means that they're more related, right? So, for example, you have the little fish and the frog, right? This first phase of embryonic development, they look very similar. They still look pretty similar, almost adulthood. I don't want to say adult, but like a juvenile. They still look pretty similar. Not even juvenile, they're still larval. All right. Then compare, for example, the frog and a mammal, right? Definitely no difference there, right? That means in their trajectory, they're much more similar because um they're similar as you go through time, right? So the longer embryos of different species are similar during development, the more closely related they are. And not just that, but you can see we are related to all these different organisms. Just look how similar we are in terms of, beginning morphology. We have a little tail, we have that little slits and that little bump here. We have that little head orientation here and so forth. So we must have some, form of similarity. Okay. Ultimately, all these vertebrates evolved from a crossopterygian fish, whatever that is. And like I was mentioning before, sea squirts are the most unlikely members of this phylum, but they do have developmental characteristics, right? If you were to get an adult sea squirt, it looks something like that. If you do a cross section, you open it up, it'll look something like this here. It actually has a little heart, a stomach, some gonads there, some little sex organs, an intestine, an anus to liberate waste, right? You have this little skin, this little integumentary system here. All right. But when you look at the larval stage, as with the other vertebrates, they all look like this. And they have this same morphology. We all have a post-anal tail. We have a notochord. Okay. We have a dorsal nerve tube. Okay. We have a pharyngeal slit. Okay. A mouth, a pharynx. Okay. These ones that are highlighted are the most important there. Okay. Very characteristic of vertebrates, which are also called the chordates, right? Uh, um, the chordates, moreover, right? Because sea squirts really don't have a backbone, right? But they still belong to this group of chordates. Okay. So anatomy, embryology. Okay. We take a look at anatomy now, right? Homologous structures. When you compare the physical form of one organism to another, right? Comparative anatomy, you basically can observe a couple of things, right? One of these things is called homologous structures, right? And basically, we have some commonalities in terms of vertebrates, right? Um, between the structure of different species. And again, like I mentioned before, it can be used as a line of evidence, right? That we've acquired these characteristics from a common ancestor, right? Now, it doesn't necessarily mean that different species can specialize into their environment, right? They can still ultimately have a limb that specializes in their environment, but it doesn't take away the fact that we came from a common ancestor, right? And this is what homologous structures are. They're anatomical structures that are common to more than one species, right? That were inherited by a common ancestor, but have different function. So same origin, different function. Okay. So from this ancestral species, we were able to create these variety of forms. Of course, every organism needs to adapt to their environment, right? To their method of living. Whales need to kind of swim about. Bats need to thrive into fly into the air, same as birds. Lizards need to walk around. Frogs need to jump around. Humans need to move about, grapple things and whatnot. But if you notice, the organization of the bones is the same. The first bone, the largest bone of the arm, is the humerus, followed by two bones here, two bones here, two little fatty bones here, two bones here, right? The ulna and the radius, followed by carpals, and then the phalanges there. The organization is the same. Same body plan, different function. Same origin, different function. Okay, these are homologous chromosomes. All right. So, yeah, in terms of specialization, it's all right, right? Lizard scale can vary from species when specialized, right? Because uh, yeah, they all organisms will ultimately specialize in what they need to do for the environment, defense, temperature regulation, camouflage, and so forth. But you will always get some homology in some structures, right? Um, and the same thing with when you compare um the leaves of plants on land and aquatic plants, although they have different things, right? Different structures, but nevertheless they have that original component, right? Petals, spines, right? Fleshy leaves or water storage, but you have a photosynthetic structure, something for support, something to photosynthesize, et cetera, right? So by having this idea, you have homologous structures, which can infer, sorry about the D there, um, phylogenetic relationships, because only organisms with a common ancestor are likely to have structures with the same arrangement. So make sure you have that idea, right? Arrangement. Okay. Arrangement there. Sometimes it could happen that structures can cease to have a functional purpose, right, for the organism. So um, you no longer need to use it, right? It's called vestigial structures. And I mentioned this in the class before, but just to kind of reiterate here, um, in the case of a whale, you have an ancestral species of the whale, which is terrestrial, had a hip bone there to support the hind legs, right? Pretty relatively large hip bone, right? But over time, that the organism kind of shifted to the aquatic environment, to the modern well, it essentially lost its formation of a fully formed hip because you really don't need a hip anymore. when you have the support of water. So it's reduced in size and it's pretty much unnecessary now, right? So these are referred to as vestigial structures, right? Some other examples for us in the human body, you have goosebumps, you have the appendix, you have wisdom teeth, right? And it could be any structure. You don't use it anymore, it's going to disappear, right? It could be bone, tissues, organs, molecules, cells. But since we still have them, you can see that there's still signs of evolution, right? Um, because they're of their presence at some point in time. Our ancestors must have used it for one reason or another, right? So vestigial structures, okay? And then you have analogous structures. Analogous structures is essentially, opposite to homologous structures. Same function, all right? Same function, but different origin. There is no common ancestor, right? The structure and the function evolved independently from in these two different species. For example, the human eye, the vertebrate eye, and the octopus eye. Same function to visualize and to capture your environment, to sense your environment, right? But the components are different. They might be similar, but um, um, their origin is different. Okay. Eyes, the octopus versus vertebrates. Vertebrate eye, the nerve fire, the nerve, ugh, the nerve fibers lie in front of sensory cells. So essentially, there's a blind spot. Okay, and I'm gonna explain that. Octopus eyes, the nerve fibers lie behind them, so there's no blind spot. Right? Developmental process is different, but it has the same function. So what do I mean by this? So let's take a look at the human eye. The human eye, basically, you have your photoreceptor cells, and the light is coming in. Whee, pew, and when it activates these little cells, it triggers a signaling pathway that would relay into the nerve fibers, right? That would ultimately run into the optic nerve. Okay. We generally have a blind spot because if you notice, there's no sensory cells here. There's no photoreceptor cells here. So there's a blind spot that we technically have. We don't see our blind spot. We can't perceive the blind spot because both of our eyes compensate for the blind spot. Okay. But there's actually a way you can test that we have a blind spot. And the reason we have the blind spot is because the nerve cells are in front of the photoreceptor cells. But in octopus eyes, the nerve cells are behind the photoreceptor cells. So they have in that sense, a better coverage than we do, right? When light comes in, it's activating all the sensory cells as much as it can, right? And then you have the signal to go to the brain and so forth and so forth. So that's what it's referring to as a example, right? Developmental process is different, but the same function occurs. There's another example, and in this case, examples are good, guys. Strongly recommend it. Wink, wink, star, star, etc., etc. Bat versus bird and bat wings versus insect wings, right? Um, insect wings are made from different material, chitin, right? Same material as the exoskeleton of certain arthropods. Birds, you have that bone, very nice cartilaginous tissue. You have bone tissue. You have the feathers to create that wing structure. All right. You take a look here. Same function to fly, but from a different source, meaning from a different origin, right? Both of these organisms could fly, but the material is different. Okay. So this is analogous structures. All right. Okay. I think that is good for this chapter. Again, like I mentioned before, I will go over phylogeny and do some practice problems in phylogeny, but essentially, that's all you need to know. Um, I do want you guys to study the fossil evidence section on your own, okay? Because that's what the task is about, and so forth. All right. Thank you guys for listening, and I'll be back with another one.